Phytoplankton are microalgae that are the base of the food web. They produce carbon through a process called photosynthesis. Very shortly, they take up sunlight, CO2, and nutrients, and thanks to a pigment called chlorophyll, they transform this inorganic matter into organic carbon that will feed other organisms. Why is this important? Well, without them, everything falls apart. So, what's your impact on it? Let's take a look and compare areas with and without people and see if there are differences in the primary productivity between them.
For each site, let's measure the chlorophyll to estimate the quantity of phytoplankton, the nutrient concentration, and the organic matter in sediment scores as an indicator for the contamination of each site. Let's go to the field and collect our samples. In each site, we have to take 3 replicates of water samples and 3 sediment cores. After, we have to analyze our samples in the lab. We start with the filtration of the water for the chlorophyll extraction and the nutrients analysis. Then we do the extraction of the chlorophyll pigments and read the absorbance at 715 nanometers and 665 nanometers in a spectrophotometer.
And finally, our results. The chlorophyll concentrations we obtained were not significantly different between the enterprise and non-enterprise sites, and the content in organic matter was only significantly different in some of the locations. About the nutrients, all the sites had the same value in the test scale. So, in conclusion, our results showed that the calculation of our chlorophyll concentrations did not show a difference between the sites, and for that, it was not possible to establish a trend with the organic matter content in the sediments. However, our lab procedures and readings may have not been very accurate.
The nutrient test was not sensitive enough to show differences, but was a solution available within our time frame. However, indications of green filamentous seaweed indicate possible nutrient enrichment in some of the sites, so further analysis should be conducted. Taking this into account, our studies did not show a relationship between the amount of primary production and inhabited areas.